And it's 12:30 on Radio Eastern Sydney, and I'm very lucky to have today with me in my studio here at 2RES FM, Dr. Jeff Simons. Hello, Jeff. How are you? It's lovely to have you here today. Today we are going to follow along the pattern a little bit. Last week I brought you Professor Peter Rowe as he was covering some of the aspects of the Children's Medical Research Foundation and what they do there. And Dr. Jeffrey Simons heads up the Oncogene Unit. Jeff, welcome to Radio Eastern Sydney, to my program Lunch Break. How about you tell us a little bit about the oncogenes and Oncogene Unit? I mean, it's all a mystery to me, except that I've given a bit of a clue. I've said that people with cancer usually go to a doctor who is an oncologist.
Right.
Yeah,
right.
So how about you tell us a bit about all of this?
Okay, well, oncogenes are genes which are involved in the induction of cancer. When a person goes along and sees an oncologist, and the oncologist says, yes, I'm sorry to inform you, but you do indeed have cancer, that's a very late event. And really what has happened over many years is that there has been changes to the cells, certain types of cells within the person's body, and those changes are somewhat cumulative. They add together, and the end result is cancer. What we as a unit, the Oncogene Unit, are concerned with is how these changes occur, how they lead to cancer, and in what way that takes place. So if we think about what genes are, genes contain the material of life, that all the information that is necessary for a person to develop normally and keep going is encoded by the genes. And it's really when something goes wrong in a certain class of genes that people are moving in a direction of getting cancer. So that oncogenes are cancer-causing genes. And if you think about that, it's a bit of an enigma. Why have cancer-causing genes within the body?
Mm-hmm. So
really it's that these genes are not per se cancer-causing genes, but they are very essential and important genes in normal cell. And we're talking about cells here, we're talking about the basic unit, the cell. They're very important in normal cellular functioning. And when something goes wrong with these genes, then cancer can develop. So it's as if a Dr. Jekyll and Mr. Hyde situation. When the genes within the cell are in their normal form, the Dr. Jekyll form, everything is okay. But when they become changed, or it's called activated, then they can assume the Mr. Hyde form and become a monster, a sort of monster within the cell that gives rise to the changes towards cancer.
Mm-hmm. You know how people, I don't know whether it's misconception or what, but people say that a cell goes wild.
Right.
Well, and overgrows.
Yes, that's correct. Mm-hmm. The cell is indeed going wild and overgrowing, and it stops obeying the normal constraints, the normal cellular behavior where cells should interact with other cells in an appropriate way. They stop doing that, and they are as if antisocial in their behavior. They become, in a way, anarchists, and they say that the cell is as if saying, I want to reproduce more of myself, and that is at the expense of the whole body.
How interesting. I never thought of it in the social context, but an anarchist cell.
Yes, that's right.
Now tell me then, the body, it sounds to me as if it is a very, very fine-tuned computer.
Yes, that's correct. All the information is there when we're born. That information is stored within the DNA in the same way that the memory of the computer is there. And it's called up as it's needed. It's called up by appropriate mechanisms. When we need, blood cells are constantly being produced within our body, billions and billions of blood cells, and that needs to be very finely regulated. And when that regulation goes awry, then problems can arise.
I see. Now tell me, what do you do on a day-to-day basis? I mean, is it long, hard, tedious work? I mean, how many years have you been involved in this kind of work?
Well, I've been involved in this sort of research. It's going towards 17 years now. That seems a long time. That also involved the acquisition of certain degrees. It's a long, slow process. You need to consider. Firstly, you need to ask the question, what do you want to attempt to uncover or discover, how to go about that, what sort of time frame is involved, And there are lots of obstacles, and you tend to stumble over those obstacles and in a sense move forward, but for every step forward there may be one or two, well, for every few steps forward there are a couple backwards as well. And it's difficult to you can't make progress in a rapid fashion because you're going into the unknown. You need to light up different areas in order to know how to proceed further. So
it's slow, tedious, demanding work because you're really looking at a minuscule part of a cell.
That's right. And as the techniques become more and more sophisticated, then that's fine, but you're getting down to a more and more basic level, and you need, in a way, to then step back and say, well, okay, I've uncovered this about a particular gene in a particular location. What does that mean in terms of the cell and in terms of the body, the person's body?
Mm-hmm. And is there much collaboration, from unit to unit or department to department or from people with other scientists overseas?
Yes,
I mean,
yes.
Is it a really sort of friendly sort of working together? Are people keen to find the answer to the jigsaw puzzle? What's the...
The simple answer is yes, that people are indeed keen to find the answer. The there is, of course, the side that there is a certain amount of competition, of course, as well, and one needs to always keep in perspective what one is trying to find, and that this is of importance to people.
Mm-hmm. So, it's a fairly isolated area to be in, isn't it?
Yes, it is. Yeah. It is. You need to realise that it is of importance. If we're going to discover what the cause of cancer is, then it needs to be at this basic level to understand what is happening in individual cells, why they go wrong, but to also keep it, as I said before, to keep it in perspective of what's going wrong in the total environment, the total picture.
Now, look, you've been working in this area for 17 years.
Right.
And give me a brief sort of resume of what sort of progression you see through the 17 years.
Well, there's been a huge progression. When I started out I was doing an honours degree in 1972.
Where was that, Jeff?
That was at Sydney University.
Mm.
The understanding of cancer was very basic, and investigators were looking at what goes on in the membrane of the cell, the outer part of the cell, and how that interacts with the environment. Are there changes within the membrane of the cell that can give you clues about cancer? Now we have progressed to the genetic level, and that's been a huge progression, looking at what goes on in the nucleus of the cell, where the genetic information is, what are the genetic changes that are involved in cancer.
Can I just track back a little bit there, because I am, in a way, I'm privileged to a lot more information than perhaps my listeners may be. So can we just go back to the membrane of the cell?
Yes.
And then we've got the cell, and then all of a sudden, to me it sounded, trying to attempt to be a listener, listening, that there was a huge leap between the discovery of the cell having genetic material within it.
No, it's been known since the 50s that the genetic material was DNA. It's been known that that's where the genetic information is stored. What was not completely understood in the 70s was that it was the genetic information that was crucial to cancer. What was thought in the 70s was that, okay, there are cells, the cells divide, we know that, they need certain genes to be expressed, but it was not understood that it was the genetic changes that were crucial to cancer development. It was thought that something goes wrong in a very descriptive way, some sort of phenomenon where the cells are no longer behaving as they should, but it was not understood that could be tracked back to the genetics. And in the last, let's say since the late 70s, early 80s, that has been the impetus for further research, looking at those genetic changes and how to uncover those genetic changes. For example, there's a class of virus which is called an Rna Tumor Virus. It's called an Rna Tumor Virus because it has RNA as its genetic material. And what is unusual about this virus, there are two things. One, that it has RNA as its genetic material instead of DNA, but the most important difference is that it is able to pick up, at a relatively rare event, normal cellular genes from mammalian tissue and other tissue, but let's say we're talking about mammals and humans in particular. As a rare event, it can pick up a gene. And what has turned out to be the case is that these genes are crucial genes for normal cellular functioning. And when the virus picks them up and introduces them to a new cell, it can actually set in motion a chain of events that will lead to cancer. So these are called Rna Tumor Viruses. They're called tumor virus because they induce a tumour. And they have uncovered this class of genes that we started off at the beginning, these oncogenes. They have been uncovered through this virus. And when one thought about viruses of this nature in the early 70s, yes, they were interesting. Isn't it unusual that they have RNA as their genome? But no one thought that this would be the first in a series of clues to genetic information about cancer.
Mm. It's tremendously interesting. And, of course, talking about viruses, I mean, everyone is terribly concerned about viruses and what viruses can do to us. I mean, everybody tells me that I meet who's slightly off colour that they're suffering from a virus. And here are you're talking to me about viruses in a really... viruses in a really sort of positive sense, that without the use of these viruses, then you cannot do your work. I mean, you can't proceed, you can't progress. So, I mean, it's really fascinating. We all think of, on the outside of the scientific world, we all think of viruses with some fear and trepidation, you know, like the plague. I mean, we're all very concerned about AIDS. Right.
Well, AIDS, it turns out that AIDS, the HIV virus responsible for AIDS, is a member of this family of viruses, these Rna Tumor Viruses. The other name that they have is retrovirus, meaning backwards, and it's that retro because they're going from RNA to DNA first. And the AIDS virus belongs to this class, the HIV virus, and it has a similar type of replication. It's closely related to another virus called human T-cell leukemia virus 1, which does indeed induce a leukemia in humans. And it's a relatively rare virus. I wouldn't like all your listeners to think that this virus is causing most types of leukemia, as that's not the case. It's a rare virus with a very defined epidemiology, that is where it occurs. But what is important is that HIV is a member of the class of virus called retrovirus. And it's if one is thinking about what these viruses do, HIV can indeed cause a type of cancer as well as AIDS, that is the Kaposi sarcoma, and some of the principles are indeed similar.
Now, Jeff, look, not so very long ago, I read in the newspaper that some fellows down at the CSIRO in Canberra had found a way to shear off a part of the gene which, you know, looks to be a major move forward in the world of science. Right.
Well, what they have found in Canberra was that it's possible to use a type of a phenomenon that was first found in plants. Excuse me, I have a bit of a cold when we're talking about viruses, perhaps relevant. A class of virus that is found in plants, and that the plants can protect themselves from this virus by a procedure that they have, which is as if cutting up the gene or the viral gene with scissors, and therefore it's called gene shearing. And this was found in the Division of Plant Industry in Canberra, and it has implications perhaps for cancer, but in particular for the HIV virus, that it may be possible to inactivate the HIV virus by using this genetic shearing mechanism.
Now, these two fellows from Canberra, one's Dr. Peacock, and the other fellow is Dr.
Wayne Gerlach, Dr. Wayne Gerlach.
Dr. Wayne Gerlach. Yes. And they tried to try and raise funds from this country, and no money.
That's right.
Could be found within this country.
That's right.
And what did France do? They leapt in and they said, we'll give you so many $1 million, and it was found within a matter of hours by a company called Le Maire Grain.
Le Maire Grain. Yes.
Which is a plant, I believe. Could you tell us a little bit about that?
Sure. They indeed jumped in and said, yes, this is a discovery that should be exploited. And it's an indictment on the political and the business system in Australia, that when a scientist makes a discovery, you can't find, no one's around. You know, you say this is something that can be exploited, that can be developed, and the attitude is, well, yes, okay, but show me that it works, or let's see some proof, or whatever. And when you're starting out, when you're just trying to do something, it's not that simple to show proof or show that other people are interested. But after the event, once the joint venture was announced, then everyone's saying, oh, well, you didn't really approach me properly or whatever. And you've got to step back. I think both politicians and businessmen have to step back and say, yes, there is a potential here, and let's put some money into it. And probably the amount of money involved, while it sounds huge to us sitting here, $20 million, huge amount of money.
Right.
But I believe that those sort of figures may be involved in an unsuccessful takeover bid for the lawyers, that doesn't go through.
Right, absolutely. But I mean, the possibility is here now that the French now are infusing all of this money into this plant genetic shearing.
Right.
And I believe they went around to all the research establishments around the place who had set up a unit such as yours, an oncogene unit, and must have been part of the condition, was it, that it be developed in Australia if it could be?
Well, at least that was preserved, that CSIRO, Division of Plant Industry, said, OK, we'll take money from outside, but one of the conditions is that the research projects will be funded within Australia, and they're looking at funding different research projects, and one of those is a project that we suggested, and it looks like they will indeed fund that, so that at least the technology is staying in Australia. It will fund research in Australia, and it will help to develop an infrastructure for young scientists to come through, to be trained, to be exposed to top-level research, and to move on. And that's what Australia needs, not to say, oh, well, gee, it can be done over in America or it can be done in Europe, because there is nothing wrong with the facilities here. There is nothing wrong with the personnel.
No.
What is wrong is the attitude.
The attitude. It always comes very low on the totem pole, doesn't it?
That's right.
And, that's really very much true of health, as coming very low on the totem pole, especially for doctors working in the area of oncology, because, sometimes and quite often the patient does not survive, and it must be a terribly hard thing for any doctor to send in the bill when the patient is going to pass away anyway, and no matter how hard they try. But of course people forget that quality of life for that person as they're living.
Yes.
Yeah, how terribly important that is.
Yes.
And, all of this fits into the total picture. And I really agree with you. I think it's so true that we do have the facilities here, and we certainly do. These French people went around and looked at these units, and they came back to you, to your oncogene unit at the Children's Medical Research Foundation, and said, Dr. Jeff Simons, we want you to have a go at this.
Right.
Now, the implications for us, Jeff, for us as human beings, you've sketched a little bit. Do you want to go into that a bit more fully?
The implications of the gene shearing?
Yes.
Yes, of course. The gene shearing is an approach which, when properly employed, the potential is to destroy the gene product before it does something bad. For example, in cancer, certain of these oncogenes that I've been referring to are not in their normal form. The normal form looks like something or other, and the abnormal form is rearranged. For example, it might be a hybrid, somewhat of a hybrid gene. And one can envisage employing these gene shears to attack the hybrid, to destroy the hybrid, so that we leave the normal gene product alone and destroy the hybrid, which causes a problem. That's how it could be used in terms of cancer. How it can be used in terms of AIDS is something like the virus is RNA. The HIV virus is indeed RNA, and it will be possible to destroy the virus using these gene shears. And that is the approach that we envisage taking, to destroy the virus using they're called gene shears.
So they literally are like scissors cutting out the nasty part.
Yes. It's hard to envisage what a little tiny scissors looks like inside a cell, but it's really a physical property of the RNA that these gene shears are actually able to, as if, twist it apart. So it's like twisting apart a soft piece of plastic or plasticine, and thereby breaking it.
Right. And so the sort of equipment and technology that you're using to do this, must be incredibly, technical. How could you describe the sort of equipment that you're working with?
Well, when you walk into a research lab, probably the first thing that hits you is the amount of equipment that's there little pieces of equipment, larger pieces of equipment, and they all have a price tag. And the price tag is quite high. But without that equipment, you can't proceed. You can't do the experiments that you want to do, and you can't reach the conclusions that you want to. So that the facilities are important. You need some sort of core facility, and then you need the additional procedure. The level of sophistication has to be such that one can grow the cells in culture, which we can do in a defined environment of temperature and acidity and so on. One also needs to be able to analyze the genes, so you need to be able to look at them. For example, the size. How big is the gene? Is it in its normal form? Is it rearranged? And you can tell that by running pieces of DNA out on, it's called gel electrophoresis, which separates them on the basis of size. You need to be able to say, is the gene being overexpressed? Is it making too much of the product? And you can do that by looking at the RNA, which comes from the DNA goes normally to RNA goes to protein, and you can see how much RNA is being produced. So you need to extract from the cells the RNA, also run it out and determine how much is being produced. So there's lots of different types of equipment. You need radioisotope to be able to analyze the DNA and the RNA. You need skilled people, obviously, that's a very important point. And the facilities, the basic facilities as well.
Is it dangerous work, Jeff?
No, not if you're careful about the constraints, the physical constraints, that you that you use appropriate isolation equipment and so on. Then it's not dangerous. No.
How many are in your team?
It's around eight people at the moment.
And then overall in the foundation, the Children's Medical Research Foundation?
Well, there's 40 scientific staff in I think it's 5 groups now, 5 different groups.
And is there a close sort of interrelation between the other groups, within, your oncogene unit?
Yes.
How does you're having constant sort of input one to the other, I imagine?
Yes, of course. The other groups are also looking at basic questions of cell biology, and there, for example, there is a neurobiology group looking at neuronal development. There is a muscle group. There is a group looking at embryo development, and each of these groups uses similar techniques that can be. We can help each other with those, and the questions involved in normal cellular regulation are also quite similar. And can be addressed in a communal way.
Jeff, you've worked in other places around the world. Tell us a bit about the other places that you've worked in, please.
Right, I've worked at the Weizmann Institute in Israel, and there I did my PhD. That gave me an idea of what research is like in America, and after Israel I went to America. I worked on the West Coast, the University of California, San Francisco. In Israel, I was concentrating more on normal blood cell development and what goes wrong in leukemia. In San Francisco, I continued that work, and I was also involved with this class of virus called retrovirus, and with looking at oncogenes. And West Coast America, people are pretty enthusiastic, and everybody wants to excel. Perhaps something that we're missing out here in Australia, that excellence is not put up on a pedestal, but in America it is. And if you want to excel at something and you want to commit yourself to that, that's okay. Whereas here it's more, okay, but why are you really doing that? So
there seems to be a real sort of ignorance within the general community towards research in general, would you say? I mean, it hasn't adopted a high enough profile.
Yes, perhaps it's partly our fault, the scientists' fault, to translate what our excitement, what our purpose in doing this sort of work is. That there is it's like going to the moon. There was something to be discovered, and it was of importance. And really, going inside the cell is no less important, and probably more important than going to the moon or going to the bottom of the ocean or whatever other pursuits mankind has been involved with. Going within the cell, finding out how a cell, which is the basic unit of life, functions, how it can keep us alive, because as we speak here and as your listeners are listening, there are millions and millions of processes going on within each person's body that keeps us alive. And really a basic question is not what goes wrong to give rise to cancer, but rather how come the body is so well attuned and has so many fail-safe mechanisms that it can indeed live in a very good state for a large number of years. That's the question we should be asking. And when we obviously with that is the question, what goes wrong? What leads to disease of different types, and what goes wrong in cancer?
That's right. That's the big $1 million question, $20 million question, huh?
Yes, indeed.
Jeff, it's really lovely that you could spare some time and come here today and share some of these profound things with us, because they truly are profound. I mean, to talk about the cell means to talk about the very substance of life. And all of us really want to go on living forever. There's no doubt about that. And the older we become, the more we clutch at the straws. And unfortunately, the more our bodies tend to fail us.
Right. I don't think science at the moment can offer immortality.
No.
But what science can offer people, can offer your listeners, can offer the people in general, is a better life, and a life in which we are more able to deal with things that go wrong with the body. And moreover, a life in which these, let's say, body failings can be found at an earlier point, rather than someone walking into the GP with a lump and saying, "What's this?" and the GP says, "I'll go to the oncologist," and he says, "Yes, it's a cancer." Rather than that, a better, something that one would be much happier with, would be the ability to find that lump before it becomes a lump. To say, yes, you've got something going on in your body, but we can give you this treatment and you won't get the lump. Mm-hmm. And therefore, it's not going to be this approach of, well, let's cut it out or use effectively poisons to kill off the cancer cells, because that has a very serious and devastating effect on the normal cells of the body as well. And a much better approach would be the one where the treatment is given before there's a
preventative.
Preventative. Yes.
Yeah. Jeff, is there anything else that I perhaps haven't covered? I mean, I haven't asked you what kinds of cells you're working on.
Well, really, cancer can occur in any type of cell. Leukemia is cancer of blood cells. There are cancers of muscle cells. There are cancers of bone cells. Any type of cell that is within the body can become cancerous. And as you said right at the beginning, what is going wrong is this cell is proliferating out of control. It's antisocial, and really any cell can do that. So the principles that we're attempting to uncover can be, in a way, applied generally to many different cell types. And that what goes wrong in leukemia probably has some specific elements, but has many elements in common with what goes wrong in solid tumours, brain tumours, or other types of tumours. And the idea of having many groups approaching the problem in different ways is to attempt to draw all this together to make it into a meaningful message that there are principles in common, and clearly there are principles which are a subset of that apply in particular to certain cells.
Mm-hmm. So you think that this, with the discovery of the genetic shears, that we have moved yet again, maybe the tiniest little step forward, but there is now much more of a possibility than what there was just a year ago of discovering cures for cancer and for AIDS, etc.
Yes, there's an important point here. The point is that if you try to say to a businessman, if I as a scientist try to say to a businessman, look, we're doing this and this, and he says to me, yes, but what are you going to show? What product are you going to produce in X years? And I say, I'm not really sure, but I think I'm going to get something out of it. And indeed, if you look at the CSIRO discovery, the result came out of a principle like that you attempt to do the research in a meaningful way, you attempt to move in a certain direction, but you don't know. It's not like making a car engine, it's not like making a refrigerator, it's not like making a radio or a record player. It's a creative endeavour, it's an endeavour of discovery, and that you really need to... There needs to be a faith in scientists, because most of the advances that have been made in the last century, the last 2 centuries, have been advances because people have been looking. If you don't look, you won't find something. And we've got to keep looking.
Dr. Jeff Simons, head of the Oncogene Unit from the Children's Medical Research Foundation in the grounds of the Children's Hospital at Camperdown. Thank you for being with us today. Thank you for the information.